professor, physician, epidemiologist. I am Dr. Sri Banerjee. I'm Dr. Sri Banerjee, core faculty for the College of Health Sciences and Public Policy at Walden University. And in this video, I'm going to talk about a very simple sort of food item, which you probably see in one form or another on your plate, which is wheat. So in terms of bread and many different types of food products contain wheat. So it's good to kind of explore how scientists infusing genetic technology and understanding of genomics have improved the wheat crop, but also go into a little bit of history as to when humans started cultivating wheat. And there's some interesting stories attached to this. So let's get started here. So wheat as we know it now, just going back about 5,000–6,000 years ago, was different and actually took some time to cultivate. Initially, the seeds would fall off too quickly, and so you wouldn't be able to actually cultivate any crops. But then currently we've found evidence 6,000 BC in Turkey, Göbekli Tepe, and some of the other archaeological sites shows evidence of cultivation of a particular hexaploid strain of wheat, and I'm going to go over the details of which specific ancestors lead to this particular strain, but it's a hexaploid strain which actually combines three specific strains to create the most perfect combination. And so recently, especially in the year 2000, there were some efforts to try to understand the complex genomics, and the hexaploidy. So what is interesting is that there's 42 chromosomes compared to other crops. This is a lot more. So if we're thinking about how many chromosomes humans have, we have 46, 22 autosomal chromosomes, and then one pair. So 42, and then 23 pairs of chromosomes. So it's 46 chromosomes for humans versus 42. So there's only four chromosomes. And then there's a lot more genetic material packed into those chromosomes. 6 times 7, 42. So it's AABBDD. And then using agronomics, you can go into understanding what parts in the gene you can modify to make the crop more resistant. I'm going to go into that a little bit more detail. So what are some of the common enemies of wheat? By knowing these, there have been certain types of resistance that have been built into the genetic material of the crop. So thinking about stem rust, UG99 stripe, leaf, and stem rust, you can see right here that the leaves are going through rust. So the genetic content and segment has been identified, and then so resistance can be built. Wheat streak mosaic virus resistance, this is kind of similar. looking at Russian wheat aphid resistance. So this is an aphid right here on the right. This causes havoc within the crop, and this can be protected against. And then wheat stem sawfly resistance. So this is a fly on the top, and so sawflies can also damage crops. And then eventually, the reason to know all of this and to study the genetic material and to understand wheat to very specifically is to really protect our food supply. And protect against food insecurity when there's droughts. These understanding the genetic components that protect against drought can help create more resilient crops, which then in turn improves also wheat quality. So if you want gluten strength, pre-harvest sprouting tolerance, you don't want the crops to grow too early, or something that just darkens fresh noodles, because color, texture, things like that make a big difference in how we perceive and taste food. So PPO is one of those that can help to do that. So in this segment, I'm going over kind of the overview of how the genome of the wheat is built, and what that means in terms of characteristics, things like that. Now I'm going to go into a little bit more detail on kind of a researcher's perspective on how all of this can make sense.